So we already know about the different types of amino acid depending upon the number of groups which they are having, that is the carbonyl or the amino group, that is neutral, acidic and basic. So one more type or classification is there of the amino acid which is dependent upon what? The synthesis, means whether it's necessary that this amino acids is synthesized in the body or should be taken externally. So on the basis of synthesis, the amino acids are of two types, essential and non-essential. Okay, so essential and non-essential, in the name itself it's there. Essential means what? It is not synthesized by your body. So obviously to get those amino acids, you have to take it through the diet. So hence they are called as what? Essential amino acids. So they cannot be synthesized in the body. Okay, so taken through diet. Whereas non-essential means they can be synthesized in the body. So some of the examples out of the 20 amino acids which you could take over here is valine, leucine, lysine, and so on. And over here in the non-essential, glycine, alanine, glutamic acid. Okay, so non-essential already because it's produced in your body, so it's not necessary that you have to take it through your diet. So out of the 20 amino acid, glycine is the simplest form of the amino acid that you could find. Okay, so then moving on forward toward the next important molecule, which is the enzyme. Now, what are enzymes? Okay, so basically, if you take a cell, there is something which is called as what? Metabolism. What is metabolism? Metabolism is nothing but the sum of catabolic and anabolic reactions. Okay, which takes place in a cell. So what is this catabolism and anabolism? Okay, catabolism means what? Breaking down. And anabolism means formation. Sum of these two is called as what? Metabolism. So in order for this metabolism to take place, we require what? Certain enzymes which will help in all the processes. Okay, whether it may be the protein synthesis or its degradation or whatever food we eat that is digested. So for all those things, we need this enzymes. Okay, so what are enzymes actually? So enzymes are nothing but biocatalyst. Okay, catalyst means those which speeds up the rate of the reaction. So if a particular reaction is going slow, if you add enzyme to it, that reaction will happen fast. Okay, so for that purpose, the enzymes are used. So enzymes are what? They are the biocatalysts, which will help to catalyze the reaction, which is taking place in a living organism's cell. Okay, so there are certain features which is very important for that particular molecule to act as what? An enzyme. Okay, so what are the properties of enzyme? Okay, first thing is they are globular proteins. So already we know two types, fibrous and globular. So these are what? Mostly the globular proteins. Then second property is what? It acts as what? A catalyst. So even if you supply that enzyme in minute quantities also, it is going to act as what? An efficient catalyst. So it's an effective catalyst even in small quantities. Third property is they are highly specific. Okay, this property I'll be explaining you further. Then fourth one is they have what? Active site. And this is the site where the reaction is actually going to take place. Where reaction takes place. Fifth one is they remain unchanged throughout the reaction. Whatever reaction they are carrying out in that particular process, they will remain unchanged throughout the process. So that is the fifth one. So they remain unchanged. The end of reactions. So these are what certain properties of an enzyme. So only if all these properties are there, it will act as an enzyme. Okay, so now as I told you, it's very important for the metabolism. So enzymes, when it is present in a cell, either it can be extracellular or intracellular. Okay, so basically, if they are remaining inside the cell and working, or from the outside, they are working inside the cell. Okay, so two things we have over here. Okay, I'll just write it down. Two types we have. One is called as the extracellular enzyme, and another one is the intracellular enzyme. Okay, so extracellular and intracellular. So extracellular means what? They will be secreted, but whatever they're working will be there, that will be externally. Okay, so suppose if it's synthesized by one cell and some other cells need this enzyme, so externally it will help in working of that particular reaction. For example, you can take any digestive enzymes. Like for example, saliva. Okay, in that amylase enzyme is present in our saliva. So initially, whatever the carbohydrates are there, while chewing the food only, it will help in breakdown of that carbohydrates. Okay, that is a starch molecule. So hence, those things are called as what? Extracellular enzymes. That is, externally, they should be exported to do a particular reaction. Whereas intracellular enzyme means what? It will remain within the cell itself and it will do its work. Okay, so within the cell, it will be present already and it will work there. Okay, so inside the organelles and the cells which are present inside that organelles, there you will find this intracellular enzyme. And the best example that you can give over here is the insulin. Okay, so these are what basically two types of enzymes and how they actually act on the process. So what happens is whenever a process is taking place, they are having something which is called as an activation energy. Okay, so activation energy means what over here? See, like whenever a molecule in a process gets unstable, okay, suddenly there is a movement and they become what? They will lose their stability and they'll become what? Unstable. So at that time, minimum amount of energy which is there that has been converted into an high energy. So that particular energy is only called as what? Activation energy. So this is a property of actually the enzyme. Okay, for example, to make it simple, activation energy of enzymes means what? For example, suppose a man is rolling a stone. Okay, and suppose there is a hill like this. So man is rolling the stone from here, is taking it upwards, and when it reaches over here, suddenly it will fall down. Okay, so here what is happening from slow speed to fast speed it's going. So this is what? A transition of energy. Fine. So this is only called as what? Transition energy of the enzyme. So suddenly when an enzyme is coming over here, it will increase the rate of reaction and the activation just busts out. The energy just busts out. This is called as what? Activation energy. Okay. So whenever there is an enzyme present, the activation energy will be what? It will be high, and otherwise it is low. That is the thing. So to explain this more easily, there is a theory or a mechanism in the enzyme which is called as what? Lock and key mechanism of enzyme. Lock and key mechanism of enzyme. This is very simple. Okay, consider a basic lock and key at your home. Okay, so you have a lock and you have a key. So each lock has a specific key. Okay, for example, if okay, this is the key for a particular lock. You consider this as your lock and this as your key. So now, if I want to open this lock, obviously this type of key only should fit into it. Correct? But if I'm using a key which is like this, just a small variation, okay, of that key also will not make it fit into that particular lock and it will not open. So the same thing is applicable over here also, the lock and key mechanism of enzyme. Okay, so for the enzyme to catalyze a particular reaction, you require one important thing which is called as your substrate, which is nothing but your lock. Okay, so when the substrate combines with an enzyme, we get what? An Se complex, which is nothing but a enzyme substrate complex. Okay, so what happens over here is when that particular substrate binds to that particular enzyme, okay, now this particular thing on the substrate is called as what? Active site. Okay, so suppose if this is your, I'll just draw it over here. Okay, so consider this as your substrate, this as the active site, this is your enzyme. So when you combine, when the substrate combines with this active site of the enzyme, what is formed is the second diagram, that is the ES complex, that is the enzyme substrate complex. And finally, when the reaction is over, what will happen is that particular product will be separated and the enzyme will be separated from this complex. Okay, so you get a product plus an enzyme. Substrate will be used up completely. Okay, so this is what called as the lock and key mechanism of enzyme. So if that substrate is not having the particular shape, okay, which is of that of the enzyme, it is not going to bind and no reaction is going to take place. Okay, so this is only called as what? Lock and key mechanism of enzyme. So in order for this any enzyme to react, there are certain factors which may affect those reactions. Okay, so what are the factors which may affect the enzyme's ability to do the process? So the factors affecting the rate of enzyme reaction. Okay, so first factor is the temperature. Okay, see, if there is an increase in temperature, whatever the molecule's motion is there, that will be fast, right? So if the molecules of that particular enzyme and the substrate, if they are moving in a faster rate, obviously the reaction will be completed in a faster pace, right? So that is the effect of temperature. So if it's low, what will happen is the reaction will be very slow. But if it is an optimum temperature, okay, that is the correct temperature at which the process is going to take place. Then the enzyme and substrate will react properly and you will get a particular product properly. So that is the effect of what? Temperature. So here what happens is heating will increase the molecular motion. So hence it will speed up that particular reaction. So ideally it needs what? An optimal temperature. So at this optimal temperature, what's going to happen is that particular activity will be more, or maximum activity will be done over there. In the same way, we have the next factor, which is the pH. Okay, so here also optimum pH is very important. Okay, so if there is a change in the pH, what do you say, level, acidic, basic, or neutral, then what will happen is that particular shape of the enzyme also will change. So if the shape has changed and it is not according to the substrate, then it is not going to bind and it will not, what do you say, run that particular process or the reaction. So pH is also the other important factor. In the same way, the substrate concentration, how much amount of substrate is present for that particular enzyme to react. Okay, so whatever rate of enzyme is there, that reaction will increase if there is an increase in the substrate. So more the substrate, more the enzymes, more the products you're going to get. So more substrate, more enzymes. Okay, then you have the enzyme concentration. So here also same, if graphically, if you want to write this, it's like whatever rate of reaction is there with respect to the enzyme, it will be directly proportional to the enzyme concentration. Okay, so these are what the factors which may affect the enzymes while they are attaching to a particular substrate. Okay, fine. Then the next one which you have is hormones. Another important thing, hormones. Okay, now what are these hormones? Hormones are nothing but certain secretions through your endocrine glands. Okay, the secretion of the endocrine glands. So basically you are having two types of glands, that is with duct and ductless. So this comes under the category of ductless glands. Okay, so this is very important to regulate your day-to-day body functions and to maintain it in a particular level. Okay, so even a small amount of enzyme, if it's produced in your body, it will give you what? A great effect on your body, or great changes you can see in your body. So obviously hormones are very important for your body. Okay, so they play what? Vital functions even if they are produced in small quantities. If produced in small quantities. Okay, so they will be having a very low molecular weight and they also will be easily diffusible. Okay, so every gland has a particular hormone or it secretes a particular hormone. Okay, for example, if we take thyroid gland, which is present near the throat. Okay, that particular gland secretes a hormone which is called as thyroxine. Now, what is the use of this thyroxine? Okay, so thyroxine basically, if it's in more amount, what happens is there happens a swelling on your neck. Okay, so that like goiter, you can say. Okay, so if it's less, if it's more, okay, it will vary depending upon how much it is produced. Okay, that is, totally depends upon the functioning of your body. Even every gland produces a particular enzyme and it plays a particular role. For example, one more we can take is the insulin. Okay, now who secretes the insulin? Pancreas. So pancreas is a vital organ if your sugar level has to be maintained. Okay, because what happens is glycogen is stored in our body or in our muscles. So glycogen breaks up into glucose and then we get what? Energy, right? Whenever we are doing a particular activity like running or whatever. So at that time, this thing occurs. That is, glycogen is converted into glucose and in turn it will give you what? Energy, right? So the insulin which is there, it is secreted in this pancreas. So it is going to increase that particular glycogen level, okay, and it will convert it into the glucose. So whenever you are doing a particular activity, a exhausting activity, so at that time what will happen is this process is going to take place and it is going to maintain that particular sugar level in your body. Okay, so making sure that low sugar levels are not prepared in your body. So these are what the role of the hormones. So hormones are basically nothing but the secretions of the endocrine glands. So if they are more or less, it may cause a problem. So it should be balanced through the diet. Okay. Then the next one which you have is the vitamins. Now, these are the another important things which are required in our day-to-day diet, even in small quantity, because they may perform a big biological function when you take it. Okay, so what are vitamins? Vitamins are nothing but the organic compounds which are required in the diet. So small quantities will be sufficient to perform a particular function because they are also important in your growth and development of the body. So a well balanced diet is important for the same. So depending upon its solubility and chemical structure, you have certain types of vitamins. Okay, so the classification. Okay, so depending upon the solubility, you have two types. Okay, that is water soluble and second one is fat soluble. Okay, so water soluble means B and C vitamins, and here A, D, E, K. Okay, so B and C are what? Water soluble vitamins. So because of that, you should consume it more. Okay, because otherwise, because they're water soluble, what will happen? They will get excreted through the urine. Okay, so because of that, the toxicity level is also low and they are not stored in your body. So you should take it on a day-to-day basis in your diet. Whereas the fat soluble vitamins which are there, they are fat soluble. Means what? They are soluble in oils and fats. So where it will be stored? In your adipose tissues, that is in the fatty liver. Okay, adipose tissues. Wherever fat can be accumulated in your body, there you will find this particular fat soluble vitamins. Okay, so each and every vitamin has a particular like boon also as well as curse also. Means if it's in more amount and if it's less amount, okay, certain changes you can see. For example, if I take over here vitamin A, okay, it's called as retinol. So vitamin A mostly you get from the carrots, fish, milk, and so on. So the main important function of the vitamin A is what? For your eyes, for the vision, as well as for the growth. But the same vitamin, if it's a deficient in your body, then what it will cause? Night blindness. In the same way, if you take vitamin B, now vitamin B is having many more classifications like vitamin B1, B2, like that. So if I take one of the vitamins such as maybe B2 we'll take. Okay, so if I take B2, B2 is nothing but the riboflavin. Okay, so riboflavin, this vitamin B2, mostly in the green vegetables you will find. So here it helps in producing certain types of enzymes. okay, which is called as FMN. Okay, so basically it helps in the redox reactions of the cells. But if there is a deficiency of the same, then it will cause a particular thing called as the cheleosis. Okay, now these things are very important in your entrance. Okay, all these vitamins, from where you will get this, what are the functions, what happens if they are deficient, which disease you're going to get. This is very important for the entrance point of view. Okay, so now what is this cheleosis? Cheleosis means nothing but you will get small, what do you say, lesions on the corner of the lips or the mouth or the tongue. Okay, so if there is a deficiency of this B2, this is same in case of all the vitamins, whatever is there. Okay, you can take any of the vitamin C, which is ascorbic acid, vitamin D, vitamin E, whatever. Okay, so here it's very important to know the source, function, as well as the deficiency, what it may cause. Okay, so this is depending upon what? The solubility. In the same way, depending upon the, what do you say, the structure of that particular vitamins, the chemical structure. Okay, you have four types. That is aliphatic series, aromatic, acyclic, heterocyclic. Okay, so here it's the vitamin C, vitamin K, vitamin A, and vitamin B. This is all on what functional group they are having. For example, if I take aromatic means these vitamins will be having some kind of aroma. Okay, so hence they are called as what? Aromatic series vitamins. Okay, so depending upon what type of structure they have, this is the classifications of the vitamins. Okay, then the next one we have is the lipids. Okay, so lipids means what? They are nothing but the glycerol and fatty acid. When together they come, then they form something which is called as what? Lipids. So these are also important biomolecule, and the most important characteristic of it is it's a non-polar solvent mostly. See, because fats, they cannot be dissolved in the water. Okay, so obviously you have to take a non-polar solvent in order to extract this particular lipid. So if you want to extract this, what you can do is you can use any of the non-polar solvent. Non-polar means what? Water hating. And polar means what? Water loving. Okay, so whatever the solubility is there, that's very limited. You cannot mix them up. Okay, so depending upon what characteristics they are having, we have two types or two classifications under the lipids. That is the complex and simple. Okay, so already we know fatty acids, glycerols makes up that particular lipids. So if I take a complex and simple lipid, the only difference is what? On the hydrolysis. Hydrolysis means breakdown. Which is going to break more easily is your simple lipids. Okay, so here in the simple lipids, they will not have an ester linkage. Now you know what is a linkage or a bond. Already in the carbohydrates and proteins, we saw the glycosidic linkage and peptide linkage. In the same way over here, in lipids, you have ester linkage. Okay, so that particular ester linkage, the simple lipids are not having. So easily they can be hydrolyzed or broken down. Okay, for example, steroids, terpenes. These are what examples of simple lipids. Whereas complex lipids means what? They will be having this ester linkage and the chains also will be very long of the fatty acids and glycerol. Okay, so they will be the esters of long chain fatty acids. Okay, so all the big lipids, for example, you can take waxes, okay, or a phospholipid. or glycolipid. Even the fat, the animal fat and vegetable oils which you use, all comes under this complex lipids. Okay, so this is all about the lipid.